Optogenetics is an emerging technology that stimulates neurons using light. Genes that code for light-gated ion channels are inserted into the DNA of a neuron, which causes the cell to be receptive to light. This technology can be used in all neurons of the body, including ones in the heart and spinal cord. But today we are focusing on the use of optogenetics in the brain. The scientific community is enthusiastic about optogenetics. The technology won the Brain Prize, and research into this field has been awarded millions of dollars worth of grants. Optogenetics has the potential to be used as a therapy for Parkinson's disease, addiction, aggression, schizophrenia, autism, depression, and other psychiatric and neurological conditions. Despite the great potential for this technology, many ethical concerns need to be addressed. The primary users are currently animal models, and in the future will be human participants in clinical trials. The secondary users are the researchers and physicians responsible for implementing the devices and determining treatment plans.
We believe the ethical concerns related to animals and humans trials should be addressed differently. For example, does testing optogenetic technology on animals strip them of their autonomy because they cannot give consent? Perhaps the question we should be asking is whether animals have autonomy. Animals operate according to their primitive instincts and therefore cannot be considered moral beings in a way that humans are. As animals do not have the same moral capacity as humans, we believe animal testing should be allowed for optogenetics. Sorry, animals.
In clinical trials, human participants are under the threat of being stripped of their intrinsic rights to autonomy and privacy, as optogenetics can access new information about their neurophysiology. It is important to address this issue as it leads to regulating the use of the technology. This brings up questions such as, to what extent are we allowed to alter the human mind? Among these ethical debates, engineers should also focus on the risks of using the technology, which are irreversible, long-term changes to the brain. The ethical considerations for researchers includes traditional research ethics, such as informed consent. When performing trials, they have an additional ethical duty to follow the principles of replacement, reduction, and refinement. It is a physician's ethical duty to ensure beneficence towards the patient by weighing the risks such as brain damage and benefits, such as treatments for previously incurable diseases, to judge how optogenetics affects their welfare. In addition, the physician must maintain the autonomy of the patient, allowing them to choose whether or not to pursue this treatment option.
Optogenetics will push the boundaries of research ethics. Specifically, what precedent will the first human trials imply? Could this ultimately lead to brain control and a complete violation of the patient's autonomy? Fundamentally, this technology will change our society's views on autonomy and privacy with access to new information. We should therefore examine some possible courses of action to address these questions. From a deontological view, the clinical trials can cause harm to another human being, and therefore they are unethical. Optogenetics can also remove individual autonomy with neural stimulation, which violates basic deontological human rights. The process of genetically modifying neurons is also irreversible, which removes a patient's right to withdrawal from the clinical studies. Proper ethical trials would be therefore difficult to conduct and therefore should be stopped.
However, do the risks outweigh the benefits of treating previously incurable diseases? It will be considered an act of beneficence from those who volunteer for the trials, whereby the potential sacrifice they make would be responsible for increasing the quality of life of others. From a utilitarian perspective, the end results remove suffering of the greatest amount of people.
We believe the best course of action would be to regulate the technology. In order to proceed with human trials, there should be a strict efficacy trial in order to minimize harm. Mind control and loss of autonomy is still a risk. However, regulations could be implemented for allowing the stimulation to be turned off when the participant desires. This creates an option for them to withdraw.
This approach aligns with utilitarianism due to the reduction of suffering. It also aligns with feminist care ethics, as it more closely recognizes that the study participants are indeed people who deserve moral consideration and respect. With regulation, risks associated with optogenetics can be minimized and ultimately, human quality of life can be improved.